of moderating the session today, or you want me to lead? How would you recommend?
Thank you, Danish. I think you should lead. It's perfectly okay.
Okay.
Go ahead.
All right. So I think this is time. Welcome, everyone. My name is Danish Patti. I'm a movement disorders neurologist at University of Nebraska Medical Center. I'm the chair for APPNA Merit Committee for 2021. It's my pleasure and honor to host another research-related webinar with a wonderful, true translational researcher. Those are very hard people to find. So grab them where you can find them. But some house rules first. Remember, a reminder to everyone that these are approved for AMA Category 1 CME activity, these webinars. So we will request our presenter to disclose any conflict of interest or any commercial conflict for any product or topic they talk about, so that you're aware of that as per this being a CME activity. These are approved for AMA CME Category 1 activity by APPNA. APPNA is offering the CME. We'll be issuing CME every quarter, every 12 webinars. So I think we're getting close to our 24th webinar, or today might be the 24th webinar. So we'll be going around and doing a second round of issuing the CME for everyone who attended as many sessions. So watch out for that, for us to reach out to you and to give you your CME certificate from APPNA and guide you for that process. And another disclosure that the CME activity cost was very graciously funded by an educational grant from APPNA RESA committee, which is the Research, Education, and Scientific Affairs Committee of APPNA, to Merit and to run these activities and program. As usual, we will go first with our speaker and then invite our panelists to give his comments, and then we'll open for Q&A. You can put your questions in Q&A box. You can put them in chat box, but they may get buried. I prefer for chat box to be used for introductions, introduce yourself, who you are, where you're from, how you'd like to be contacted and reached out. Put in your chat box any other comments related to the topic or links or anything that you would like to share. And we prefer that you put your question in Q&A box. That'll be easier to do. And then if we have time, then like usual, I would be happy to allow you to make your comment and give you a microphone. So raise your hand if you'd like to make a comment or introduce yourself, and we'll give you a microphone to make your comment towards the end as part of the discussions. With that, let me start with our topic for the day. We will be talking about translational research, very, very critical element for success of clinical healthcare enterprise in any country. So translational research, today we will be talking about research in bone growth disorders for clinician. And we have an excellent speaker, a thorough researcher, Dr. Farasat Zaman. Dr. Zaman is at Karolinska University Hospital in Stockholm, Sweden. Dr. Zaman completed his PhD in medical sciences with specialization in pediatric endocrinology at the Karolinska Institute in Stockholm and postdoctoral training at the Hospital of Sick Children in Toronto. Dr. Zaman then joined the Karolinska University Hospital as assistant professor and currently working as senior scientist and supervising PhD students. So Dr. Zaman holds numerous prestigious awards, including Young Investigator Award by International Pediatric Foundation, University of California, Marie Curie Scholarship, HK Princess Lovisa of Sweden Award, and highly competitive fellowships from Swedish Medical Research Council, Swedish Research Council, and Swedish Childhood Cancer Foundation. Dr. Zaman has won Nature Reviews Rheumatology Award of January 2017 issue for cover page image for establishing organ culture systems of human growth plate cartilage. He has published in prestigious journals including Nature Medicine, New England Journal of Medicine, Lancet Oncology, Scientific Reports, Leukemia, Clinical Orthopedics and Related Research, Cancer Research, Endocrinology, Bone, +1, and Annuals of Rheumatic Diseases. With that, let me hand over this microphone to Dr. Zaman to make his comments and share his screen for the presentation.
Thank you very much, Dr. Danish, and everyone from the APPNA organizing committee for offering me this, allowing me this opportunity to speak from your forum. And this is amazing platform. And I was already discussing with Dr. Danish. I think this is brilliant step taken by APPNA. And so today I'm going to show some of our translational projects which are designed basically to move forward the finding from bench to the clinic. And we will discuss also what are the models that can be used by clinicians without going into extensive training, and then continue their research. And we will also share different clinical questions and where do we stand and what we are doing here in Stockholm, Karolinska. So I would like to share my presentation here now. Yes, so yeah, as you can see from the topic, basically this is very much about translational research for not for clinicians but also for others in biosciences, focusing on bone growth disorders. Primarily, we will discuss here in children, but of course we can also open up discussion or talk anything question which arises about adults. So I'm affiliated with Karolinska Institute from Stockholm and with the Department of Women and Children Health. And this is my email, which will be shared later by the end as well. So if you have any questions later, you want to get in connection with us, you can use this email. So this is the Karolinska Institute building here in the new hospital, and this is the new building as well, you can see here. So there are some modifications going on with the hospital. So no potential conflict of interest with the data I will be sharing. So before we move on to the questions or our topics, I would like to show you, this is the picture of the Karolinska Institute. So this is the main entrance of the Karolinska Institute, and as a medical professionals, you are all of you are aware of this, that the Karolinska Institute is also known for the Nobel Prize in medicine and physiology, and this is the building. This is the Nobel Forum, which is just located here on the left side as you enter the Karolinska Institute, where the Nobel Assembly sits every year, and they look at the discoveries in physiology and medicine and nominations sent to them, and then they decide about the Nobel Prize. So if you make any discovery or something in any field, you will definitely come here and get your prize. So the toxicity of the therapeutic drugs, clinical challenges and the translational research. We know that with the drugs which are widely used in the clinics, they do have some toxic effects as well, and some of those effects are quite significant, but we do not have the options basically other than to continue with those drugs. And one of the important area of the research is to how to further minimize those toxic effects of these fantastic clinical drugs, and how to make the life of the patients more safer and thereby improving their quality of life. So the key questions in our research group that we are addressing is the toxicity of the therapeutic drugs, how these drugs have the toxic effects on different tissues, especially on the bones in children, what are their long-term and short-term effects, and also the intermittent use, how the uses of these drugs, for example, glucocorticoids, they affect the bone health, and also how to assess the toxicity of these drugs in different organs, and then based on the knowledge or information that we gather from these objectives, how to develop new treatment strategies to prevent this toxicity or the side effects of these drugs. So a little bit discussing about how the inflammation in neuromuscular disorders, we know that the pro-inflammatory cytokines like TNF-alpha, IL-6, IL-beta, and many other cytokines, they are upregulated, and they have effect both systemically and locally within the tissue. And to suppress this inflammation and to treat the neuromuscular disorders, one very widely used drug is the glucocorticoids, different types of glucocorticoids. They're widely used in the patients, both in children and in the adults. And amongst other effects, side effects of the heavy use of the glucocorticoids, beside infection and loss of appetite, it's also the poor bone health. So these glucocorticoids, they suppress the growth in children, they can reduce the bone mineral density in children, but also in adults. And adults, mostly this is the osteoporosis or the reduction of the bone mineral density. Similarly, another example is the cancer, different types of cancers where we are using conventional chemotherapy, novel therapeutic drugs, as well as radiation. And in this situation, you see that beside this is the long list of the side effects. Again, the bone health is also affected. So in the cancer patients, we know from the clinical data, also from the preclinical data, that the cancer survivors, they suffer from bone fractures throughout their life. So this is one of the biggest problems. And so the use of the glucocorticoids in children, both short-term or the long-term, they not only enhance the cellular toxicity, but they also impair the bone health. So primarily affecting the growth of the children in fast-growing individuals, but also triggering the osteoporosis, both in children and in adults. So in this paragraph, you can see the bone growth curve and the fractures probability in children. So here, this is the growth curve of a girl who had asthma, and she was given inhaled glucocorticoids from the age of 8, and she continued until 13 years of age. And as you can see here, that in the growth curve, gradually her growth was reduced, and which indicates that the long-term use of high-dose glucocorticoids, they have a negative impact on the growth in these children. And this is also in this slide here, in this here figure B, you see this is from patients with the Duchenne muscular dystrophy who are heavily dependent on the glucocorticoids. And as you can see here, those who are dependent on chronic use of corticosteroids, they have the higher fracture probability compared to non-chronic corticosteroids. So, so we cannot avoid, even, I mean, there are effects, but we cannot avoid, or we cannot stop using these steroids or glucocorticoids in these patient groups. So the question is that how to prevent these side effects, or how we can improve the overall the effects of the drugs, desired effects. So before I show some data, I would like to give some brief description about how the bone growth is mediated. And as you can see here, this is the picture of the long bone, and each long bone basically consists of the growth plate at both ends. Here you see as blue line here. So this is the growth plate cartilage. So this growth plate cartilage is responsible for the bone elongation. And this bone elongation and the mesenchymal stem cells, which we know that they are differentiated. They differentiate into osteoblasts and adipocytes and chondrocytes. So the chondrocytes primarily are located within the growth plate, and these chondrocytes, they proliferate and differentiate and thereby take part in the bone elongation process. But the mesenchymal stem cells also differentiated into osteoblasts. They also contribute in the bone formation, strengthening the bone. So this is just very the brief introduction about the bone growth. So this is the picture of the growth plate, a little bit more microscopically how it looks like. If you take the picture of the growth plate and if you look at the under the microscope, the growth plate, you will see that the growth plate has the chondrocytes. So all these cells are chondrocytes. So the growth plate is very unique in the sense that it has only one type of cell population, which is the chondrocytes. And these chondrocytes, they are at different differentiation stages. And the first zone is the resting or the stem cell-like zone. This is here you see. There are very few cells, but they are a critical population in the growth plate. If experiments have shown that if these cells are depleted or if these cells die, the bone stops growing. we also have shown some data previously that by using proteasome inhibitor, which is used in the cancer, if these cells they are killed, the bones they don't grow at all. So these stem cells they give rise to proliferative chondrocytes, and these proliferative chondrocytes then they become hypertrophic chondrocytes, and ultimately they die. So this is the natural phenomena happening. So if there is any irregularity or inhibition of the proliferation or differentiation within the growth plate, this directly affects the bone elongation in the long bones. So the proliferation and differentiation is important. Cell death and increase in the cell death plays a crucial role in the impaired bone growth, as well as local and systemic growth hormone and IGF-1 regulation also affects the growth plate and thereby affects the total bone elongation. So, so clinicians are play a very important role actually when investigating these effects in children, because they are the ones who observe these effects in children, and so if they can take part in the investigational studies, that makes the whole the project much more productive and much more targeted and beneficial for the patient. So one aim of my talk is to introduce some very simple translational methods for clinicians and others who are not even clinicians, but they want to do some projects in the field of bone growth. Without using using a lot of money or resources, you can still perform pretty good experimental questions. And just briefly, you see that these are the primarily three more working models that we use. One is the in vivo, where we use mice, rats, and rabbits to investigate the effects of different drugs on the bone health. Then we have the cells or cell lines like stem cells or the chondrocytes or osteoblasts. So they can also be used. But then there's also a third model, which is called ex vivo models or the bone organ culture system. So this model, the bone organ culture system model, it's stronger than in vitro, but it is just in between in vitro and in vivo, because here you can study how the bone is bone formation ex vivo. You can also study the bone growth ex vivo. You can also see how the bone, the different drugs, they affect the bone ex vivo without affecting the animals. So we'll go with a couple of clinical questions. So for you just to understand this, that how we are addressing these questions, and I have put this first question is that how to minimize or block the side effects of an anti-cancer drug vismodegib on bone, and why this is important. If you look at this paper, which was published in OncoTarget, so vismodegib basically it's a hedgehog inhibitor, which was approved to treat medulloblastoma in children, and this was a fantastic drug, and but after approval, the clinician they started using it, but then they found that this drug basically is causing permanent growth plate fusion in some children, and which was a big concern, because this means that those children who are treated with the vismodegib, they will never grow. They will remain very short because their growth plates are destroyed. And this is the title here. You can see here the irreversible growth plate fusion in children with medulloblastoma treated with the targeted hedgehog pathway inhibitor. So this was a big shock for all for the clinicians and also for the patients, because the Roche had to stop this, and as I mentioned here, that these findings resulted in a major trial amendment to restrict the drug to skeletally mature patients. So then they decided that this drug will not be given to children who are not skeletally mature. So it means that who have not reached to the puberty. So after puberty, so then they can use it because the growth plates are fused by that time. So so we had this. So based on some of our data that we had been working on hedgehog inhibition and the bone growth, we actually searched for some possibilities how to prevent this the effect of the hedgehog inhibition on the growth plate. And in this study, which was published in Scientific Reports, we showed that the radial shock waves, when they are applied on the bones by using an ex vivo culture bone model system against vismodegib, which is a clinically used drug, we can prevent the bone growth suppressive effects of the vismodegib. So this was very well taken by scientific reports, because this was some kind of like a big hope for everyone. And we know, obviously, we are about to start with the in vivo experiments so that we can move on. But this was a very important preliminary finding. So what we did is, now I'm gonna show you here with some, introduce you with the methods as well, because this is the right time. So to address this question, can radial shock waves, which are already used in the clinic for different purposes, can they prevent the effects of the vismodegib on the bones? So for this purpose, we used a bone organ culture system, which is very well established in our group. We are widely using it. And in this model, what we do is that we take the rat embryo at embryonic day 19 to 20, then we do a microsurgery on paws and remove the small bones, metatarsal bones from the paws. So everything has to be done under the microscope, because they are very, very small. And then this is how the bone looks like once it is isolated from the paw. And then we transfer these bones individually to each well. So under cell culture media, defined with the defined cell culture media that you can go and check on our web in our publications. And interestingly, these bones, they grow just like they grow in vivo conditions. So they increase their length. They also have like whole chondrocytes activity. They just behave, they respond to growth hormone, they respond to IGF-1, they respond to glucocorticoids, they respond to all those drugs which normally they respond under in vivo conditions. So this is a fantastic model to test locally that if the effect of your drug, how this effect is mediated on mediates the effects within the growth within the bone. And secondly, importantly, in the absence of any systemic factors. So these bones, you can grow these bones in the absence of any systemic factors. So it does not contain any growth factors. It's just BSA and then sodium glycerphosphate and beta glycerphosphate and some other reagents. So and then once we put these bones, transfer them into each well, we expose them to the different drugs. For example, in this case, it's a vismodegib, which is the hedgehog inhibitor, and then we also applied the Shockwave on these bones. And then we take the pictures of these bones every second day. So and follow them for 1 week or 2 weeks. You can follow them for 4 weeks even. And then we make a growth curve of this bone and see this, that how bones respond to the drug, investigative drug. This is another ex vivo model where you can study, because here you're studying bone elongation, right? So how the bone grows longitudinally. But if you want to study the bone formation, then people, they have shown this, that you can remove the calvaria bone from this embryo and then grow this similar way in the petri dish, and then and apply the drug of your interest or whatever you want to investigate, and stain them later on for the bone formation markers, and or even do the qPCR for the bone formation related genes or growth related genes. So these are two very interesting models here. You don't need heavy funding for this. So it's a very cost-effective method and very reliable, actually. And you can address very nicely the questions by using these models. And also, especially when clinicians who do not have the time, they can use these models very easily in their spare time, and if they want to investigate anything on the bones. So the application of the bone organ culture system, it's now very widely used, actually in pharmaceutical industries, also in preclinical CROs, if they are screening small molecules or the peptides or new drugs, investigational drugs, before moving on to in vivo studies. Because going to in vivo study directly from cells, it can be quite costly. It's not ethical, because then the health of the animals is also very important for us. So therefore, doing anything on the bone organ culture system before moving to the in vivo study saves our time, efforts, and resources, and also good for animals. And so you can study very with confidence questions related to growth, bone growth, and osteoporosis by using this bone organ culture system that I just showed in the previous slide. You can also do preclinical toxicological studies of new drugs, as I mentioned earlier, like steroids, peptides, hormones, cytostatic drugs, and their direct effect or a direct crosstalk between these drugs and the bone, especially if you want to study the direct crosstalk in the absence of any systemic factors, growth factors. And then also do as a proof of concept study, environmental pollutants can also be used on this model to study how the environmental pollutants may affect the bone health in children and in the other populations. So coming back to this, some data that we published, that how we managed to reverse the growth retardation caused by the wismodegib. So this is the growth curve of the bones cultured ex vivo. And this is the bone length in percent from day 0. And the bones were treated for 5 days with the wismodegib, which is the hedgehog inhibitor used in the cancer. And as you can see here in the red line, the bones, they were growth retarded, significantly growth retarded compared to the control, which is the black here. So we used quite low dose of the wismodegib because the higher dose would completely kill the bone or cells, and the bone will not respond. So we titrated the doses, and we found that this was the best dose that can show us the growth retardation, as well as the bones are also continuously growing. And you see here that until day 15, the bones are growing. And then we actually applied some bones which were treated with the wismodegib. We applied Shockwave on those bones in the petri dish, and until day 5, and then we followed them, their growth. And as you can see here in this growth curve, that the bones with the wismodegib and exposed to the Shockwave, shown here in the green line, they actually they didn't display any growth retardation, but rather they started catching up back to the control level, and it was a significant rescuing effect in these bones when we applied the Shockwave. So this was a very good and exciting news for us. And then we look further to confirm the growth velocity as well. As you can see here, the growth velocity was also improved in these bones which were exposed to Shockwave in combination with the wismodegib. And this is the histology of the bones. As you can see here, that the bones with the Shockwave, they showed stronger chondrogenesis. So we thought and we proposed like a hypothetical model. It's not still in the clinic, but that's what we need to do in the in vivo studies that we have planned now. That if we apply the Shockwave at different locations in those children who are receiving or who receive, for example, wismodegib for the medulloblastoma treatment, but at the same time, if we apply Shockwave at different locations of the long bones where the growth plates are located, perhaps we can actually prevent the toxic effect of vismodegib directly within the growth plate, and thereby there will be no interference even with the cancer therapy, because shockwaves also by itself has anti-cancer effect. But here it will be more targeted at the growth plate level. So one may ask this question that if I use the bone growth organ culture system, how close this is to in vivo, or like in children, or like in animal, because this is still an ex vivo model system, or it's a bone organ culture system. So just for your information and knowledge, I just took this graph, this we published earlier, where we have shown the growth curve of the bones which were cultured ex vivo. And they were treated with the dexamethasone, the glucocorticoid. As you can see here, as expected, the growth dexamethasone induced growth retardation in these bones. And these bones, they were still growing, but with showing the growth retardation compared to the control. And this is the growth curve of the girl who had received steroids for a long time. And you can see that this growth curve and this growth curve, they are very close to each other. Although the total bone length would be different because these are small bones. And here, this growth curve is obtained from a child where the systemic growth factors are also present. But here, this is the absence of the growth factors. So what I'm trying to make the point here is that this method, the bone growth or bone organ culture system, is a very strong model to study the growth, bone formation related questions. If you have a new idea, or if you have some new concept that you don't know how to refine this, I think this is the best model to start with it. It requires only a few hours for you to go into the lab and take care of the bone, look at the bones, apply the drug, just take the pictures, and then you can analyze the data later on wherever you are, so you don't have to worry about it. So if we move on further to the clinical question number 2, that how we actually are investigating here in Stockholm, that the glucocorticoids, which are very widely used in pediatrics, and especially, for example, in Duchenne muscular dystrophy, and we know that in those patients, the bone fractures and the growth retardation, these are also very severe side effects from these glucocorticoids, and also in inflammation. Again, this is the same slide I have shown previously, but just to show you again this, that we know that that steroids or glucocorticoids, they cause growth retardation or suppress the growth in the children, as seen here in the growth curve, but they also increase the probability of the fractures in the treated patients. And unfortunately, we do not have very reliable methods or adjuvant treatment strategies, something that can prevent these effects which are induced by the steroids, especially when they are given in high dose concentration and over a prolonged period of time. And yeah, so this is the same, that that since we know that the glucocorticoids, they have the growth suppressive effects if they are used over a long period of time. And the key questions are that what are the better treatment strategies for patients, and whether this is intermittent treatment to be given to the patient, or if it's a short term. But then beside this, although the clinicians have worked hard further refining the treatments for these children, but the focus is also to develop new glucocorticoids and analogs which are less toxic, and they do not cause the growth retardation or induce osteoporosis in patients. Although still there is, we are not successful yet in developing the these effective analogs, but there are also attempts if we can combine these glucocorticoids with something else which can reverse or prevent these toxic effects on the bone health in the patients. So keeping in mind or thinking about this strategy, we have actually explored a small peptide to reverse the effects. Before showing that data, we have previously published in PLOS ONE, where we did a very interesting experiment, and based on the data that we had, we thought that the BAX, which is a pro-apoptotic protein, it is widely, it's a very well-known pro-apoptotic protein which regulates different effects within the cell, including apoptosis and also proliferation in the cells. So we had some indication that the BAX basically is highly upregulated when the cells are exposed to the glucocorticoid. And based on this idea, we knocked out the BAX in the mice, and we treated them for 4 weeks with the high-dose glucocorticoid. So this is the growth curve in the male mice, and you see this is the black one is the control wild-type mice, and then the red one is the and the red one is the wild-type mice, which had received, which was given high-dose glucocorticoid. And you can see here that these animals, they displayed severe growth retardation after 4 weeks. But when we knocked out the BAX from the mice and gave them same dose of the glucocorticoid dexamethasone as given here, these animals, they did not show the same level of growth retardation as seen here. This means that, and the similar trend was seen in the females as well. This means that the BAX basically was playing some key role in inducing the growth retardation. So based on this data, then we started wondering, is there anything that can suppress the BAX by pharmacological treatment? Because in patients, you cannot knock out this a gene that you want. But maybe using small molecule or maybe peptide or something, if we can combine it with the dexamethasone, thereby suppressing or bringing it down the activity of the BAX, perhaps we can reverse the effects of the glucocorticoids in the growing individuals. So then we screened different peptides and molecules, and we found humanine, a small peptide from the literature. Humanine was discovered in 2001 from a Japanese, by Hashimoto and his colleagues from Japan. This is and they labeled it as a neuronal rescuing factor because it was detected in the neurons originally. And this is a 24 amino acid small peptide and widely expressed in heart, skeletal muscle, kidney, liver, brain, and gastrointestinal tract. And some preclinical studies have shown very promising effects in Alzheimer's disease, type 2 diabetes, and in stroke. So we investigated that whether if we combine the humanine with the glucocorticoid dexamethasone, what would be the effect on the bone? Because humanine is also known to bind with the BAX. So it basically blocks the activity of the active BAX. So based on our previous data, where we had shown that the BAX knockout animals, they are protected from the toxic effects of the steroids, can we improve the bone health or bone growth in mice if we combine the dexamethasone or steroids with this small peptide human? So then we conducted this long study, which was published in FASIB last year, with the title that the humanine is a novel regulator of the hedgehog signaling and prevents the glucocorticoid-induced bone growth impairment. So first, by using same bone organ culture model system that I explained earlier in previous slides, we found that the humanine was very effective actually to reverse the growth retardation caused by glucocorticoids. Here is the growth curve of the bones cultured ex vivo, and you see here in the red is the growth, is bones treated with the dexamethasone or steroid glucocorticoids. And when these bones were co-cultured with the humanine, these humanine reversed the growth retardation caused by the steroid. So based on this preliminary data, we moved on, further continued with in vivo testing in mice, in young mice. So these mice received glucocorticoids for 4 weeks. We collected the X-rays every week. So the bone growth was monitored from day 0 till end for until week 4. For total duration, it was 28 days. And so they had received daily injection of the dexamethasone alone and in combination with the humanine peptide HNG. And as you can see here, again, this is the growth curve of these mice. This is the increase in the femur length from day 0 after treatment. And you see here in the red line that steroids, as expected, they caused growth, induced the growth retardation in these young mice. But when we combined the steroid dexamethasone with humanine peptide HNG, it completely reversed the growth retardation caused by the steroid. And And then this was also similar effects were seen when we looked at the total body length. So this was a very interesting observation and exciting for us that we were able to reverse the effect of the glucocorticoids, which are normally very toxic for the growing individuals. But one question here is a very big and key question that remained, that we know that the glucocorticoids are used as, to suppress or to treat inflammation primarily. And what is the effect on inflammation? Because if the combination of this peptide with the glucocorticoids interferes the desired effect of the glucocorticoids on inflammation, then there's no point of using it. There's no sense to further continue. So then we addressed this question as well. And there we treated the cultured human macrophages to induce the inflammation in these macrophages. We treated them with the LPS, and you see here in response to LPS, they showed high TNF alpha excretion detected in the culture media. But when we treat them with the dexamethasone here in the yellow, as expected, the dexamethasone suppressed inflammation TNF levels in these macrophages, which is expected, right? But when we looked at the level, cytokine level, TNF levels, in those groups where we had combined LPS with HNG or LPS with dexamethasone and HNG, similar anti-inflammatory effects were seen. So there was no interference from the peptide by itself against the desired effects of the glucocorticoids. Even actually the peptide by itself, if you look at this group, LPS and HNG, 10 nanomolar here, and then also at 100 nanomolar. The peptide by itself had a similar anti-inflammatory effects as seen with this dexamethasone, so which was very encouraging and very good news for us. And we further validated this anti-inflammatory effects in vivo in mice, which was treated with LPS for 3 hours, and to trigger the cytokine reaction. And then we collected the serum from these animals and we measured the IL-6 levels in these mice. And as you can see here, a similar effect as it was seen here in human macrophages, we found similar effect that that humanine by itself was also anti-inflammatory. And when it is combined with the dexamethasone, it does not interfere with the desired anti-inflammatory effects of the dexamethasone. So then we actually also we have developed another technique in our group. It's a culture of the growth plates. You remember that I showed you the picture where the growth plates are located. They are on the both ends of the long bone, and we wanted to develop a small method where we could grow the human growth plate ex vivo, because there are sometimes cases where we get the growth plate from patients, but nobody had ever actually thought or tried to culture these ex vivo, these growth plates from children ex vivo. So how do we get these growth plates? So this is the scheme you see here. Basically, this is from the epiphysiodesis operation, where the children who are with the leg length discrepancy, for example, if the one leg is growing too fast compared to the other, or in conditions of tall stature, where the one child is growing very fast and then the child wants to stop the growth, then these children are referred to the clinic, and the orthopedic surgeon, and together with the pediatric endocrinologist, they decide to remove the growth plate from the long bones. And this is how it is done. So the growth plate is destroyed at the long bones. But instead of destroying the growth plate, we give a special needle to the orthopedic surgeon. So he inserts the needle and extracts the growth plate from those bones instead of destroying them. And then we collect them back. And this is how it looks like, the growth plate obtained from a child after the epiphysiodesis. And then we cut them into small pieces and grow them ex vivo in the Petri dish. So this paper has already been published. And when we took the image of the growth plate from this child, we were able to detect the stem cell zones, proliferative zone, and hypertrophic zones, which I showed previously picture, which was taken from mice. But this was the first ever demonstration from a human growth plate showing 3 different zones of cells, which included resting zone, proliferative zone, and hypertrophic zone. And the Nature Reviews Rheumatology, they selected this picture for their cover image. So can we use this model, for example, to detect anything if the glucocorticoids have any effect on the chondrocytes or on the bones? Because normally you cannot investigate this in patients. And whatever findings we have, they are from mice or from rats, and one may always put a question up there that these animals, they have different bones, they grow different, they grow at different rate versus humans. How to translate this? So this model was very effective for us, where we could investigate the effects of the steroids, for example, like the dexamethasone, the glucocorticoid we had used. So we exposed these biopsies to the dexamethasone, and we found that after 24 to 48 hours, these growth plates starting showing signs of dead cells. That's apoptosis. So which had actually never been demonstrated before, that the glucocorticoids, such as dexamethasone, can trigger apoptosis in the human growth plate of a child, obtained from a child. So this was also new information. We learned that these cells are very sensitive to the glucocorticoids. And then later on, using the same model that we established here in Stockholm, we characterized a couple of cancer drugs as well, for their effects, potential effects on the bones before they are tested in the animals. So this model is also one thing that can be used by the clinicians if they have access to the growth plates. Then we also actually tested that if this humanin peptide that we had previously used to reverse the effects of the glucocorticoid on the growth, whether this can also prevent apoptosis in condition of arthritis, which are treated with the glucocorticoids. As you know, that the glucocorticoids are widely used in arthritis as well. So and apoptosis is one of the biggest also problem in the growth plates and also in the articular cartilage in conditions with arthritis. And as you can see here, that the apoptosis was increased in the growth plates of mice with arthritis when they were treated with the dexamethasone. And when we treat them with the dexamethasone plus HNG, the apoptosis was suppressed. And this is in the growth plate of the arthritis model, and this is in the apoptosis was apoptosis in the articular cartilage in mice with arthritis. So showing this, that the peptide, when combined with the glucocorticoids, it prevents apoptosis even in conditions of arthritis. We have just I'm not showing you some more data because it is under revision, where basically we have shown that when we combine the peptide with the glucocorticoid, it does not interfere with the clinical scoring of arthritis. In other words, the desired effects of the steroids, they remain same when suppressing the arthritis in mice. It's not in humans. But that data will be soon available So then one of our colleagues, Professor Princess Cohen from University of California, who had been working with us for years, and so he actually is very much interested in mitochondrial peptides, and they have tracked humanine. As you can see here, that they treated the mice for 33 months, which is a lot and a very long time duration just to see this, that what happens with if you treat them for a long time with the humanine peptide, what happens to the mice? Are there any toxic effects? Because humanine peptide seems to be very promising in different pathological conditions. And you see here in the curve, it's the percent survival, and this is the months here, and you see here that that there was no effect, toxic effect of super high, very high dose. They had used quite high dose of the humanine that compared to what we had been using, but they did not find any noticeable side effects or toxic effects. Interestingly, when they looked at the measured the humanine peptide levels in the monkeys, and they found that the peptide levels, they are decreased with the aging. So this suggests that perhaps the peptide has the effect, is also regulating aging as well in the tissues. And there are a couple of questions that we are addressing now, but we don't know yet how this effect on aging is regulated. So the 14-month treatment with humanine peptides showed no toxic effects, no severe side effects. Rather, it actually showed an improvement in the health span related parameters, like, for example, decreased in IGF-1 and trend for decrease in the leptin in the mice. And humanine levels, they also show this, the humanine level decrease with the aging in the monkeys. So this is now we have just got some money from European Society for Pediatric Endocrinology for the long-term treatment of mice with arthritis, combining this humanine peptide with the glucocorticoids, and see this, that if we can move on further with this. So a little bit summarizing this, the glucocorticoids plus humanine and shockwaves, what I have shown, are this something like a potential new treatment strategies? Are they directing us towards some new concepts or new treatment strategies to improve the bone health and to prevent the toxic effects of steroids or glucocorticoids, and also the chemotherapy? So what we show so far is that the glucocorticoid treatment, it impairs the Hedgehog signaling in the growth plate of the chondrocytes, and interestingly, it also, the glucocorticoid treatment also suppresses the local humanine expression within the growth plate. I didn't show here because of the limited time, but if you go and look at my paper on published in FASIB, you will see this, that how the glucocorticoids, they affect the local humanine expression within the bone. And that humanine treatment, it restored the Hedgehog signaling, and it prevented from the glucocorticoid-induced bone growth impairment in young mice. And also the systemic treatment with humanine prevented the apoptosis in chondrocytes in mice model of arthritis, which is also very good news when we want to take further the peptide into different pathological conditions of bone pathological conditions. And from data from Professor Cohen from California, they confirmed that there is no side effects of the peptide, even given at a very, very high dose, and when they followed the mice for the 14 months, but it rather has beneficial effects like improvement in the health span parameters. And we also using this bone growth organ culture system, where we have shown this, that the shock waves, when applied, they can prevent the toxic effect of the vismodegib anti-cancer drug, and which actually shows us very promising results, whether it is possible to prevent the growth plate fusion in children who have stopped basically now, which this treatment has been stopped in children, although it's a very effective drug, anti-cancer drug, but unfortunately because of its effects on the growth plates and the bones, it has to be stopped by, and FDA has asked to stop this in growing children. But now soon we will start this in vivo experiments where we will apply the shock waves on the bones in rats or in rabbits and see that if this can be in vivo can also the same effects can be achieved in vivo as well. And then interestingly, shock waves are non-invasive, which further adds up actually the clinical significance of this project. So it's a completely non-invasive method to improve the bone growth or bone health. So we are also actually investigating scoliosis, how the growth plate is affected with scoliosis. And if anyone, clinicians or anyone who is interested, please contact us. We can start some joint projects. So this is a very brief, short glimpse of our activities, what we are doing. So we are primarily looking at the bone health and bone growth, both looking at the osteoporosis as well as the growth of the bones. We are looking at inflammation in conditions of arthritis and IBD, neuromuscular disorders like, for example, in Duchenne muscular dystrophy, where the glucocorticoids are widely used. We are investigating, one of my PhD students, she's investigating the combination of the prednisolone with human peptide given to MDX mice, which is the neuromuscular dystrophy mice model. And she has found quite interesting data so far. It's not published yet. We are also affecting how the glucocorticoids and cancer, so for example, in ALL or neuroblastoma, they impair bone growth, and how this can be, health can be improved, bone health can be improved in these conditions without compromising the effects on the cancer. We also investigate growth hormone IGF-1, signaling, humanine, selective estrogen receptor modulators as well, signaling as well. And then we have recently started in orthopedics field, looking at the role of growth plate in different conditions. For example, leg length discrepancy, or in leg length difference, also referred as a leg length difference, how the growth plates, activity in the growth plate chondrocytes is impaired in those conditions, and can we reactivate the activity in those ones. And here, so we have a collaboration with Royal Institute of Technology in Stockholm, which is a very exciting project. And we have already two PhD students. One is orthopedic surgeon, and one is the pediatric endocrinologist who are driving this project. And finally, I thank to all these colleagues, including Professor Savindal, who was my supervisor and also a group leader here in Stockholm, Professor Pinchas from California, Klaus Olsson here from Gothenburg, Professor Bent Fadil from Karolinska Institute, Hemal Mehta, Bettina, she works now with AstraZeneca, Yunhan Zhao, he is an MD PhD student, but currently working for PhD under my supervision. My another student, Zheng Pei, he's an orthopedic surgeon. Lily Tim, who is a pediatric endocrinologist from Switzerland, he's also now working towards his PhD in a leg length difference project. Katya and then Therese Siderwal. So with this, thank you very much, and I'm now ready for the questions.
Thank you so much, Dr. Zaman. Let me invite our worthy panelist. We have a pleasure of having Professor Ali Java available as a panelist to comment. Professor Java is a diplomat of American Board of Internal Medicine, American Board of Internal Medicine Endocrinology section, and Board of Nutritional Sciences. He is MD from US and MPH from US, and is working as a consultant endocrinologist and a medical director for WillCare in Lahore, Pakistan. Professor Java, your comment and its potential trans.
Thank you very much, Dr. Zaman, for an excellent talk. Excellent, really enjoyed it. It was a treat, an intellectual treat. Just have a question that what I gather is radial shockwave therapy is extremely safe, and it has been used even in adults. And I think you have published some studies as well, other than the one that you have mentioned. Clinically speaking, in glucocorticoid induced growth disorders in children, do you think just giving prophylactically radial shockwave therapy could have any benefit? Has it been studied, or do you think it has any merit?
So thank you, Dr. Professor Java. And actually, I have met you actually in Karachi, I believe, in one of the meeting, and it's nice to meet you again here. It's a very excellent question, actually, and we never actually have looked at this from this angle, that if the glucocorticoid induced growth retardation can be reversed with the shock wave. And I mean, if we compare with the vismodegib, which is very, very toxic, it's a hedgehog inhibitor, compared to the glucocorticoid like dexamethasone, which is not that toxic. So I would not be surprised that if we see any positive effect of the shock wave on when reversing the effects of the glucocorticoids. But we have not looked at this because we are more focused on the peptide-based approach, where we are combining the glucocorticoid with the peptide, and this peptide by itself has anti-inflammatory effects. But this is a brilliant question, so it should be investigated.
Professor Java, my question to you would be that being a practicing endocrinologist in Pakistan, what role and opportunities you see for such kind of translational clinical research in endocrinology or bone growth or something, and what challenges you see in Pakistan?
The biggest challenge that I find in Pakistan is because the centers take a lot of time to establish, and many times people are not encouraged that much to pursue that, neither financially nor tenure. So that is one of the biggest challenges. In order to accomplish something like this, you need a sense of security. You need at least Maslow 3 or Maslow 4 level of support, which many a times the government sector cannot provide, and private sector doesn't have enough push to do that. That is one of the big things. To do it logistically, it's not that difficult. As you've said, we have very well established animal sciences departments in Punjab, in Sindh, and I have a feeling that you procuring the right kind of animals and harvesting them and getting the bones and all these things can be done. It's just the person who is going to be done, he needs to be supported very effectively, and that is a big challenge.
Dr. Zaman, I'm curious if you want to just summarize what kind of work or collaborations you're doing in Pakistan and what opportunities you have found to do this kind of translational research in Pakistan.
Well, I mean, I have actually some ongoing collaborations with Quaid Azam University in Islamabad. And so I have tried to redirect their research towards translational. For example, they're the Department of Chemistry. So they are brilliant people. They are sitting there. They have the knowledge, they have the technology to develop something new. But how to translate those things to the patient, to the clinic? And maybe with finding out the right gap of, where is the gap of knowledge? And for example, they were developing polymers, right? So we actually, we have now together developed a small polymer at Quaid University, which can be loaded with the dexamethasone. And then you can actually release the dexamethasone with very low electrical stimuli, like with very low voltage, very low voltage, just to trigger this and it will release the dexamethasone where at your point of interest. And then it can also release the dexamethasone in response to low pH in the surrounding tissue. As you know this, that the cancer cells, they have low pH in the where they grow compared to the normal cells. So in that, in such kind of strategy, we can, I mean, trying to move on further. This is one example. And now, unfortunately, I have not been there for the last 2 years because of the COVID, but I'm very much open from other medical faculties or from biosciences, if they want me to establish some models there with them. I think this bone organ culture system should be very seriously considered, because this will improve not only the research in the bone and orthopedics, but also in endocrinology, where we want to investigate the effects of different hormones direct effects on the bones, medicinal plants, how they affect the bones, that can be investigated. And yeah, so definitely I'm open. So either they contact me through APPNA Merit forum or directly they write me, but I would definitely love to help the local research groups to establish this.
Wonderful. I see at least one person's raised hand, so I will get them in the loop on discussion. One quick question. Somebody raised this question, what do you think is the mechanism of effectiveness of the shockwave therapy? And I was wondering, is that technique can be applied to other growth cells or other therapies besides bone growth or cancer cells or something like that? Is there other potentials that you want to mention?
Yeah, this is Dr. Samia Taslim, I guess. She asked this question, and I think this is I mean, when we look at the literature, shockwaves are known basically already to exert anti-cancer effects as well, even in cancer, different cell types, depending on the level of the resistance of by the cancer cells. The shockwaves also have been shown to promote the bone formation. And you have to find the right dose of the shockwave, how long you have to treat. The mechanism, there are widely different mechanisms reported so far that the shockwaves can also enhance the angiogenesis in the related genes. What we found actually is the mechano-signal transduction signaling pathway. So the mechanoreceptors are highly upregulated in response to the shockwave, which is quite obvious. But we are also now looking at the shock waves and their effects on beta-catenin signaling and hedgehog signaling. So these are some potential pathways.
Wonderful. Dr. Samia Taslim, please go ahead, introduce yourself and ask a question or make a comment.
Yeah, thank you. Give me opportunity for APPNA. I'm Dr. Samia. I'm PhD in microbiology, University of Karachi. I develop one research group working of different natural medicine. Recently, we start some work for the cancer treatment from marine-based products. So we search out we have the very good lab structures. If any sir is collaborate for any project, we are open. Our Pakistan have the good resource for the plants and marine products. Those have very good anti-cancer activities. To show we have the pharmaceutical microbiologists and pharmaceutical scientists with us to give the better steady plan in future.
Thank you, Dr. Singh. Dr. Zaman, if you also want to type your email address on the chat box so people can
definitely welcome, Dr. Samia. You can always write me, and whatever support I can provide, I would be happy and give even intellectual inputs wherever needed and with the projects. And here is my email. I'm typing here in the chat box, so please do not hesitate by writing me email, and we can discuss the different projects and especially if there are any clinicians listening this or those who want to work in the field of bones growth or bone health related questions. So definitely we can look at the project, we can look at the feasibility by some joint small funding from Sweden as well as from HEC and from other sources.
And I think a potential collaboration for future clinical trials for like that polymer that's being developed in Qaid-e-Azam, those are really interesting ideas, and there should be, I highly encourage everyone to think about it. And if you have a lot of patient population that are related to this or know someone who has it, please get them in touch with Dr. Zaman for future clinical drug development. I think that will be hugely beneficial. I see a lot of questions in the Q&A box. I don't know if you have access to Q&A box or not. I can pick questions, or you can see if there is a particular question that is of high interest. We're over time, so maybe we can only touch another 1 or 2 questions before we have to close for today.
Yes.
Anything that seems really interesting or appealing that you want to comment on?
Here I have, so if you can kindly forward me, Dr. but the emails of this from this chat box, because some people they have written emails regarding communications for the projects, but I'm just looking at if there are any.
There's a Q&A box separately, Dr. Zaman. I see questions about other steroids besides dexamethasone having impact, aerobic exercise, effect on development of humanin production, role of humanin in neurodegenerative disorders. Do you use specific microscope, confocal or light microscope?
I can, yeah,
for
I
studying?
can quickly go through this because I was looking at the chat box
Yeah.
previously. So Dr. Ali Asar, he says, is there any adverse effects on bone health dependent upon inhaled steroids? Yes, depending on how long you use and what doses do you use and what is the age of the patient. So definitely inhaled steroids, they do have effect, even especially in children, as I showed this in asthma. Where they actually had effects on the growth. But of course, there's a lot of data available, you can see there. Then does aerobic exercise has anything to do with this peptide humanin production, and exercise do retard is the commercially available? This is a very interesting question. So one of my colleague who is also an MD, so they have looked at the effect of the exercise in diabetes on regulation of humanin in the body. So they found this that when you perform exercise, the humanin peptide level is increased in the body, and thereby it also brings down the glucose, the sugar level in the body. So that was quite an interesting study. That is also shown, has been reported. So that, yes, so exercise does affect the peptide levels, humanin peptide level in the body. Question: if dexamethasone has bad effects, then why would you give to the premature, especially before delivery? So I mean, I am not a clinician, but here I can just at least tell this, that because sometimes when the children are, like premature born, so the dexamethasone or glucocorticoids have to be given to induce the differentiation in the lungs and also in other tissues. So the clinicians, perhaps Dr. Bhatti can comment more on this later. So the glucocorticoids are very, very important and very, very important line of treatment available so far. Do you use confocal microscopy or light microscopy study apoptosis? We have used both confocal and light microscopy to study the apoptosis in the growth plate. Is there any way of evidence of humanin in neurodegenerative condition like Alzheimer's disease? I reckon it might be beyond the scope of your talk. Some evidence of humanin in there. Yes, I mean, there are a lot of studies where they have looked at the humanin and its potential effects, therapeutic effects in neurodegenerative disease conditions. For example, it has been shown that a single injection of the humanin once in week, it improves the cognitive functions in the mice. But all the data is in preclinical studies. It's quite interesting and impressive peptide. Is it a naturally occurring peptide or just a synthetic compound? Yes, it's produced by the body, by all cells. The peptides, this is produced by the mitochondria. So all cells, they produce it. But what we have used, it's a synthetic peptide in which we have induced a mutation in one amino acid. So changing one amino acid in the native peptide makes this peptide 1,000 times more potent. So we have used a synthetic peptide in our study. Can inhaled steroids affect bones of the fetus? Can inhaled steroids by asthmatic mother affect bones of the fetus? I think this is a clinical question, and a clinician definitely must answer this. But when looking at the preclinical studies, when the pregnant mothers, mice, are given steroids or high-dose glucocorticoids, that affects their offsprings as well. So that is one thing. Do you have any data on other steroids like dexamethasone is used? No, often clinically used. Really nice. Thank you. Yes, we have the data by Mohammed Usman Amir. We have the data on prednisolone, which is used in the clinics, right? Very widely used. So that data has to be published very soon. Is there any way of reversing effects of glucocorticoids in teenagers who use them in childhood for asthma? That is a very difficult question, and I'm afraid probably not. We have not looked at this. Thank you very much.
Let me invite Dr. Saeed Uzair to make his closing comments and close the session for today. Dr. Uzair, please go ahead.
Thank you very much, Danish. it was very enlightening talk, and I think that's the objective of bringing the research and promoting the translational research. And I'm really very excited to know about the work of Dr. Zaman and association with Quaid-e-Azam University. And I think we are exactly following the objective of merit, which is the research, and this is the talk is quite aligned. And I think the comments are that a lot of good things are happening with the diaspora from the Pakistan, and this platform was very much needed to bring all this work to one level. So that's phenomenal, and thank you very much, Dr. Farasat, for sharing your thought-provoking work, and where the Nobel Prize is going to be dished out. So that's really interesting to know. And I think we will continue, and we look forward to have the more meaningful relationship with the other researchers. So thank you very much indeed, and look forward and wish you all the best.
Thank you, sir.
Dr.
Thank you very much. Thank you.
Dr. Zaman, would love to have you as part of our merit research subcommittee. I'll reach out to you and see if you have availability. Thank you so much.
Thank you, sir.
Thank you, everyone. Have a nice day.
Thank you. Thank you,
Thank
Jay.
you.
Hafiz.